Our next graduate speaker is Genela Morris, who was a student of Haggai and who is now senior lecturer, I suppose, at Haifa University. And you remember the year? What year did you finish?
That's a very good question. I think, yeah, third.
Anyway, and maybe this is an opportunity, first of all, to apologize for the fact that some of us have to leave in the Middle East when there are really many things going on in parallel here. And also to all those outstanding graduates, we really had this choice problem that too many of our graduates, scientists, too many are doing excellent work, and we had somehow to balance it. And I really apologize, especially for my own students who are not here, but are still part of this community, and we'll do it again later. So, Genela, please.
Thank you. So thank you for inviting me. I think fifth year is the answer for your previous question. So I will talk today mainly, well, about a couple of different projects that we're working on, but I will present mainly one that talks about representation for learning, for decision making, and how it is molded by adaptive needs or by reward. And so I will just start with the people who are now in my lab. I've been at Haifa for the last 3 years, so I accumulated some people in the lab. I will tell you about one project that relates to the hippocampus and how it can serve as an internal model for many different processes, including learning of different sorts, and including decision making, of course. Then I will go back to Striatum, which is where I started with Haggai, and try to tell you a little bit about how these two structures, the striatum and the hippocampus, interact, and this type of learning that you will see that sort of requires both. And I think I will not talk about at all about the last part, which is our latest addition to this whole concept of representation learning. So these are just the people. Yael and Od are postdocs in my lab. Tuba is a PhD student. Sonia and Rui are master's students. Sudeep is PhD students. And as you see, all male members of my lab refuse to send photos, but they are male. And Dana is my lab manager. So what I started with back in the 5th year of the ICNC, working on with Haggai, was neural correlates of reinforcement learning. And so what we did there was work with monkeys. We recorded from dopamine neurons, from striatal neurons, from all kinds of different neurons, and we tried to figure out or find out just a bit more about the correlates of this very basic and very important type of learning, which basically talks about the ability to map between a certain state of the environment and an action that will yield reward rather than an action that will yield punishment or will reduce reward. So this is the field of reinforcement learning, and very luckily we know a lot about it from different types of disciplines. So it was first introduced in psychology, and the psychological concepts of the formal type of learning, the formal concepts of this type of learning have been put forth at the turn of the previous century, I guess, by Thorndike and others. And we also know a lot now about the computational elements that might take place there, of different algorithms that are taking for this type of learning, because again, it is a very ubiquitous type of learning, and it's very useful because there's no real feedback. There's no real reinforcer. There's no elaborate error propagation. So it's really good for many type of natural and unnatural learning tasks, and also people in the world here in the audience, and maybe a bit in the podium as well, have contributed quite a lot to how exactly this type of learning is done in the brain. So the common thought right now is that do you have a pointer? That state information is conveyed to the striatal circuit from the cortex, and this striatal and basal ganglia circuit right here performs the actions that will eventually yield the reward, and that is modulated by synaptic plasticity mechanisms, by LTP and LTD in a differential manner, it doesn't really matter, which is modulated by responses of dopamine neurons coming from the midbrain. So we know quite a lot about this type of transition, where it's done and how it's done, except we're missing one fundamental problem, and it is, what is the state exactly? So what in the whole input that comes from the entire cortex can we define as the state that needs to be learned about, needs to find the correct actions, be modulated by dopamine, etc. And so if we take an analog from medicine, because well, many students were formerly medical students, the part we all know so much about is the part of trying to find medication for some diagnosis, for some known disease. But actually a much more difficult task, and I think 8 or 9 seasons of House MD can prove that, is the fact of first forming a diagnosis, of taking the entire list of symptoms, trying to filter out which are relevant, which are not relevant, and form them into one particular state that should be learned about. And then the mapping between state and action becomes almost trivial. So in different projects that we're working on, we're trying to find out both conceptually, theoretically, and also in terms of the brain mechanisms and neural correlates, how exactly is this representation learned? Is this representation really relevant for behavior, and is it modulated by behavior, by relevance of behavior? And I hope I will be able to convince you that it is. So the first project I will talk about is mainly done by Tuva and by Od, and just to show that this is really not new, the whole idea of sub-sampling the inputs of the environment for learning, and again for decisions, is as old as at least as this book by Tolman, who wrote in his very famous book that we'll see another citation from soon. He wrote that although a rat, he's talking about rats, rats and men, a rat is bombarded by a lot of stimuli, the nervous system is very selective as to what it will let in into any given time. So it really does make sense to try and look at this reduction and representation in various aspects of the brain. But what this paragraph is more famous for is this description of Tolman about how learning and navigation can take place. And what Tolman describes here in his model is the fact that an animal does not just learn stimulus response association, but rather it forms some sort of an internal model of the world and of the relationships within that world in the brain. And this internal model, Tolman termed a cognitive map. And the term cognitive map, that again in Tolman's words referred to, I think, any type of internal model that an animal might use, was taken by O'Keefe and Adele in 1978 to describe what is going on in the hippocampus. And the reason he did that was these very famous type of responses that you get from recording in hippocampal brain cells. So if you record, and you will hear more about in other exotic animals, but if you take a rat or a mouse and you record single neurons from the hippocampus, you will see this typical response, which means the neurons will fire in certain locations in space and will not fire in other ones. And this is usually the way these things are shown, and we will show them here. So I'm sure you are familiar with this description. This is a rate map of one single neuron in the hippocampus. And this area here that's highlighted with brighter colors, meaning that this neuron fired more in this area in space, is called the place field of this neuron. Okay, so neurons in the hippocampus show place fields. They show specific locations, and they map the entire environment. And this is the reason why back in 1978 they proposed that the hippocampus could serve as a cognitive map, meaning a literal map of spatial representation on which a rat can navigate. Just two aspects of place cells that have been repeatedly shown and described and that are very important for us right now is well, two features. One is the fact that they're very stable. So if you take a rat and record its neuron in one location, take it out, come back, and you can come back days, weeks, months later, the same cells will represent the same space, the same location in space. So their representation is very stable. However, if you change the room completely, or if you change very salient features of the environment, the cells are said to remap. So they will code, again, locations in space, which again would be very stable for that environment, but will be completely different, and at least to the best of our knowledge, completely random in relation to its coding in the previous environment. So we can use this feature of place cells and talk about the state the hippocampus or maybe the rat believes they're in by this phenomenon of remapping. So if in two situations the cells behave the same or they're stable, it would mean that according to at least this cell, the state has not changed in any significant manner. Whereas if a cell remaps, or if the entire map remaps, it means that conceptually, for this ensemble or for this hippocampus, or I would argue for the animal, the state has changed. So we can use this phenomenon, record from multiple single neurons in rat hippocampus, and look at which situations the cells remap and which they don't, and trying to figure out some common principle in defining the representation in this internal model by the cells. So this is what we do. We take a rat, we implant it with multiple tetrodes, which allow us a pretty good way of recording many single neurons with good quality at the same time. And now we take the animal, we put it in this very, very simple task. So they're inserted into a ring maze, which is basically just a linear maze that is closed. And they're entering at some point, let's say, they're entering the maze here, and they have 2 minutes to run and collect their rewards. The reward is given here. They, in different conditions, they have different cues around, but they always have olfactory cues lined in the ring maze that would indicate the location of the reward. So the rat enters, runs, gets the reward, we take the rat out, we rotate the maze, we put the rat back in, and he basically goes and gets its reward. All this time we're recording neurons, and now we want to ask how spatial is the coding in the population of hippocampal neurons that we see. So what you will see here is 10 such trials, and these are the different odors. This would be the rate map, and as you can notice here, the place field of the cell is somehow pretty close to the red odor right here. And as the trials progress, or not, the timer is going, that's good. So as they progress, you should be able to see that the behavior of the cell follows quite closely the red odor. Now, I should note right now, because this is a bit deceiving, this is the location of the odor, it's there for visualization, that's the example I chose you. You have those cells all around the maze, so they're not preferentially centered around odors. So we're trying to quantify that, so we just correlate the rotation angle, which is just an angle of some arbitrary point in odor space, and the best angle of the neuron, and we compute the circular cross-correlation. This is depicted as linear for visualization again, and this is the cell I just showed you, which rather than coding just of space, coded olfactory space pretty nicely. And actually, if you take the population of recorded neurons, this is the picture you get. Most of them code olfactory space very well, rather than something else, or rather than not coding anything maybe. So we were trying to find out what is different in the cells that coded that space and the cells that didn't. Did they code something else? Don't they code? Are there possibly two populations, each coding a different reference frame? There is a bit of talk about that. So we tried to look at those, and luckily we looked chronologically at our cells. So we started with the first cells recorded and just went on. And what we saw very consistently was that at the beginning of the training stage, so in the first couple of days, maybe a bit more, what we see, rather than what you saw before, is something similar to this. So the place of field of the neuron is now somewhere here, between the red and the blue, closer to the red. And it just stays there in geographical coordinates. So in sort of GPS kind of coordinates, or visual coordinates, or maybe some other stimuli that the rat may know about. And this is the cell we saw before. So no matter how much we rotate the maze, the cells keep firing at the same location in real space. And so if we try to see what the animal is doing at the same time, we can see somewhat of a abrupt shift in the animal's behavior. So we're trying to quantify this behavior. The behavior is very disorganized. We're not making the rat run in one particular location. We're not imposing anything on the rat. Moreover, the rat will get its reward even if it runs in the wrong direction, or even if it runs really slowly. And yet there are many correlates that let us think that there is really something that goes on in this case at about the age 6. In different rats, it's somewhere between 5 and 7. And so the first thing you would notice is the running speed. The running speed goes down. This is the average running speed across 2 minutes. Rats know where they're going. They get the reward. They stop running, or they go very slowly. So that might not be surprising. What is surprising is how long it takes them to get to the stage. If we try to look at the rats trying to learn their environment, explore the environment, again, you see a very abrupt reduction in the number of times the rat was just exploring, changing its direction back and forth. And what I think is more impressive is this shift in the number of corrective turns, so U-turns that the rat did, finding one, reaching the first odor, figuring out that this is not the shortest way to get to the reward, making a U-turn, going back and collecting the reward. So again, this rat has learned at a very, very, very slow pace that it should rely on its olfactory modality rather than on anything else to collect rewards. And now if we look at the activity of the neurons at about the same time, and we try to quantify what the neurons are really coding at each time, at each recording date, we devise this index, which basically goes from -1 to 1. When -1 says that the neurons, or most of the neurons, code geographical cognitive map places, where at 1, the cells would code olfactory space. And if you try to plot this index across time, you would see that the rats really go from coding some general geographical coordinates to coding odor coordinates, and again with a very similar time coordinate what I described before. And this is, since this learning is so slow, it gives us a rare opportunity to figure out what's going on in the hippocampus during learning or formation of a cognitive map. Because usually when rats are introduced into a visual maze and they keep running around, maps are formed within minutes, a small number of minutes. Whereas in our case, both behaviorally and neuronally, this takes many, many days. And therefore, it gives us a very good opportunity to figure out pharmacologically and physiologically and in any other way that we can what's going on here. And so just as one example, this is from results from Josh Burke in the hippocampus. What he shows is a rat entering some maze. He sees this is a power spectrum of different frequencies in the LFP, and what he shows is a very, very short, 1 minute long, robust peak at the beta frequency, which he feels is the correlate of the learning in the hippocampus, of the spatial learning in the hippocampus. And what we find is, again, a very prolonged beta period in our data. A very prolonged beta period, which means that, again, we have a very long window of opportunity to see what's going on. So to conclude here, this is just what I've been trying to show you, that maps, overlaid maps with many different features, are impossible to navigate in. And it really depends on what you need. And if you're just starting to be a graduate student and you want to know where to study about the brain, because that's what you want to do, then you might look at a feature map with accentuated academic features, which will tell you that Haifa is the only place that has 2 universities that teach you brain studies in Israel. But it might also tell you that Jerusalem is the only one that has an ICNC, and you might want to go there. But as you get older and you realize that academia is not all it's cracked up to be, you might try to move somewhere else. So in the 1 minute I have left, I will just say that the next stage that we're looking at is whether this in itself is also reward dependent through dopamine. So midbrain dopamine neurons have very sparse, but they have projections to the hippocampus, and LTP in the hippocampus is dependent on dopamine much in the same way as it is in the striatum. So it would be very intriguing to see whether same responses of dopamine neurons shape this very slow rearrangement rate of hippocampal map. And the way it could do that maybe is by increasing the occurrence of what are called sharp wave ripples, in which, and this is really, really quickly, but in which sequences of activation of neurons are replayed. So this is from Lee and Wilson, I guess, but again, it's been shown again. Sequences are replayed and condensed in a very, very narrow time that is good for LTP, STDP type of mechanisms. And what we see is that through learning, the occurrence of these ripples is much larger. So the next thing we want to do is to see whether learning, the occurrence of ripples, replay, and the remapping change with manipulations of midbrain dopamine pharmacologically, optogenetically. And I think I will have to stop here. So.